Welcome, everybody. Good morning. It's my pleasure today to introduce Dr. Stephanie Huang, who is an associate professor in the Department of Experimental and Clinical Pharmacology here in the College of Pharmacy. And she is also a member of the Genetic Mechanisms Program that Masato Yamamoto and I co-direct, and she's a very active member. We were able to steal her away from the University of Chicago in 2017. And in this very short time, Stephanie really has made a big impact on the research here in the Cancer Center. Let me tell you a little bit about her career. She received her BS degree from the Shanghai Medical University, and then went on to do a master's in clinical pharmacy and drug metabolism at Purdue. Then she got her PharmD equivalent at the National Association of Boards of Pharmacy, and went on to do a PhD at Purdue. And that ended with a certificate in clinical pharmacology. So she's actually a board certified clinical pharmacologist, but then found herself doing more and more research and delving into big data sets and analyzing clinical practice. And so she is really a driving force here at the university in the Institute of Precision Medicine. And one of the major focuses of her work is to analyze high throughput data and integrate that to study drug sensitivity prediction. And when Stephanie joined the faculty here, she started out and was very active in breast cancer. And in the meantime, she has branched out into prostate cancer. And I know she has a very strong ongoing collaboration with Scott Dame. She is very well funded by NIH grants and foundational grants. And so today she will talk about bridging preclinical drug screening with patient molecular profiles for biomarker discovery and drug repurposing. Take it away. Thank you for being here.
Thank you, Anya, for that very kind introduction. Talking about my lab, really, like Anya said, the main focus is what I think of are two words: translational pharmacogenomics. As you heard about my sort of training background, I really I was lucky to get exposed to many different research areas. So currently the model my lab has adapted on is really this integrate from bench to computational to clinical research together with the goal to study pharmacogenomics. And this is my laboratory's website. So I always consider us lucky who were conducting cancer research in the time that there are large-scale cancer data sets with rich molecular profiles available. One example that we probably all of us have known and heard about is the Cancer Genome Atlas project, the TCGA. As of today, the TCGA has already had over 12,000 tumor sample banked, and they are representing over 33 different tumor types. And for every single sample in TCGA, multiple layer of molecular data, both on the DNA, RNA, microRNA, epigenomic, protein expression, very rich data are publicly available under TCGA for these over 10,000 samples. And TCGA is not alone. There are many other similar type of data sets, focus on pediatric cancer. ICGC is the International Consortium for Cancer Genome Consortium. This is extension of TCGA to the international scale, and many others, all of which has done extensive genomic profiling and other molecular profiling for cancer samples. These data really make a huge revolution in our understanding of the molecular basis of cancers. If you can recall some of these early publications in the high impact journal, you can envision there's some sort of heat map about sort of redefining cancer based on new molecular features, and then followed by a Kaplan-Meier curve figure 2 to show these newly defined molecular phenotype, molecular subtypes are associated different survival outcomes. I thought all these are really cool, but my work is very much oriented around drugs. So one sort of lacking I feel from reading these papers that it's so nice that we are able to now define cancer molecularly, and knowing that different subtype of the cancer are associated with different survival outcomes. But what can we do? Can we prescribe better medication for those subtypes that have showed poor prognosis? So we decide, in the current time, we really want to take advantage of existing data. There's so much to play around and so much to learn from. So our strategy has been taking advantage of these data, use or build computational tool to understand the relationship between molecular features and drug response. And we then experimentally validate these results, and also at the same time collecting clinical sample with ultimate goal to translate the finding from the sort of hypothesis generated through a computational method and validate in the laboratory, and eventually validate them in clinical samples. So we've talked about all the cool things that these large data sets, cancer data sets have. There's these molecular subclassification, but one thing made it really hard to study drug response is that the majority of these data sets, drug sensitivity information or even drug treatment information are missing. So that really feels like a missing piece of puzzles in order to sort of linking these rich molecular data to drug response. As a solution, so we look beyond the clinical. So all of these representing clinical data sets, these are tumor sample obtained from patients, but we all know it's unethical and impossible to treat individual with, as a drug screening tool. Nobody can be screened with hundreds of compounds, but this is routinely done on the preclinical models. So they are large-scale preclinical drug screens that's available. But what I'm showing you on the screen, this is one of those examples, the genomics of drug sensitivity in cancer. So this is an initiative started by the Wellcome Trust Singer Institute. Basically, they screen so far about hundreds of different drugs in close to 1,000 cancer cell lines. And for every single cell lines, they also, in parallel to those TCGA type of data, DNA, RNA, and methylation and microarray data are publicly available in these cancer cell line model. And PDSC, the genomic data, drug sensitivity in cancer, is not the unique effort. Here in the US, the Broad Institute has also started Cancer Cell Line Encyclopedia. CCLE, again, is to characterize molecular background of about 1,000 different cancer cell lines. Moving beyond CCLE, Broad has conducted the CTRP, which is the Cancer Drug Therapeutic Portal, which actually also screened, as of today, about close to 700 different compounds in these thousands of cell lines. And this is not only true in cell lines, it's also preclinical drugs, high throughput drug screening has also been done in patient-derived tumor xenograft samples, so the PDX. This is an effort, I believe, led by Merck. They actually did, so they screen, again, hundreds of different compounds in these PDX models. Again, for every single PDX tumor, they know the detailed molecular features. So that triggered our thoughts about, well, in the preclinical data, we have these molecular features. We also, let's see what I can get the mouse to go. So in the in vitro model or PDX preclinical model, we have the molecular features of every single cell lines or PDX model. We also have pharmacologic traits, which are measured drug sensitivity. Our thought is that can we build computational model to learn the relationship between the genomic features and pharmacologic traits, and then apply this relationship matrix to additional sample that contain the genomic feature. So in here, when I say genomic feature, it could be DNA, RNA, or any other things. Then that would allow us to impute potential therapy in those additional data sets. So really, that's our goal, to learn relationship between molecular features and drug response in the preclinical setting and translate that to the clinical or other samples. So that brings us to the first application. I mentioned we want to learn the relationship. Again, we started building model using one of the two, actually both of them, the Sanger and the Broad high throughput drug screen. Using machine learning, we were able to build relationship matrix between transcriptome, so gene expression in the cell lines, and hundreds of different drug sensitivity. So for every single drug, you can imagine there's an equation that's the output of it is the relationship between every single gene and the drug sensitivity. We then apply this model to additional samples. So these additional samples could be additional cell lines or PDX or animal models or clinical samples that has the transcriptome data. Then we can impute drug sensitivity. Once that's generated, we can then do either biomarker discovery or drug repositioning. For example, the reason I say that, imagine in TCGA, there are over 10,000 samples. So for every single sample, we were able to impute hundreds of 700 different drug sensitivity for every single piece of TCGA tumor. And in TCGA, we have all sorts of molecular data, copy number variation, mutation, expression, and methylation status. We can do sort of a genome-wide association study between the samples and drug sensitivity for biomarker discovery. For the time, I'm not going to have a lot of time to talk about biomarker discovery, so I will focus on drug repositioning. So with that, that's the sort of the ongoing work. We continue to work on these CRPC patients and samples. Our goal is to better nominate candidate drugs that can be used to treat these metastatic castration-resistant prostate cancer. As Anya alluded to, we're also funded by NIH to do work in breast cancer. We have actually generate candidate drugs, and in that area, our drug has already, we show efficacy in animal models, and we're considering getting into the clinical trials for the top drug that we're nominating. And we recently started a new brain cancer collaboration with Dr. Toga and Clark Chen, as well as some work in the pediatric brain area with Dr. Logan Spector and a very talented fellow, Robert Galpin. And we have started some work in ovarian cancer and F1 mutated cancer as well. So all of these areas, we can relatively quick nominate, using our computational model, we can nominate different drug candidates and there are different stages of validation ongoing. So with that said, let's move on to the second part of my talk. So far, everything we talk about are focusing on nominating candidate drugs one at a time. So what we all know in cancer therapy, oftentimes we use combination therapy. And this is not only, it has been shown very high efficacy. Many of the cancer treatment are indeed combination therapy. And that also gives sort of a better opportunity to inhibit the development of resistance. So there's theory about by giving multiple drugs in combination, we could better control the eventual relapse and recurrence of disease. So people are trying very hard to see, can we nominate drugs as the most efficacious combination? So the work conducted here is by the very talented graduate student and now postdoc in the lab, Alex Lane. So our approach I will share with you is through a novel mechanism called independent drug action. So a little bit, yes.
Stephanie, just real quick, if you could, it sounds a little muffled, so if you just maybe get a little closer when you're talking, I think it might pick up your volume a little bit better.
Sure, I will, sorry.
No, thank you.
Okay, so this is the mechanism. So thinking about, again, back into drug development, specifically in, so our traditional pipeline of study developing a drug, usually it goes from preclinical, once that test is done, move into phase 1, phase 2, and phase 3. But in the preclinical model, usually, you screen compounds in the cell lines. But thinking about, the example I gave you early is this monotherapy screen, preclinical screen. Let's say we want to test 100 drugs in 1,000 cell lines. That will result in about 100,000 experiments. So that has already been done by GDSC or CTRP. So these data exist. However, the same scenario when you move into combination, if you want to test 2 drug combination for 200 drugs in 1,000 cell lines, that will result in close to 5 million experiments. And that's not even talking about 3 drug combination, that's much higher numbers of experiments. So, currently our technology, the high throughput drug screen, would allow us to do things focused on the monotherapy level, but it becomes technically infeasible and not to do for those drug combinations. And actually for the 2 drug combination, NIH has launched this Almanac project, which basically tested 5,000 drugs in combination in 2 drug combinations in about 60 cancer cell lines. But still, that just only touched the surface of what we need to test without any hypothesis. So technically, this is infeasible to really exhaustively test every single drug combination in preclinical setting. And in addition, this whole traditional pipeline of drug development, the success rate has been really poor. There's a report about only 3.4% of drugs starting on the preclinical stage get to the eventual FDA approval. So we really need to do better on that. So our hypothesis is that it is possible to create a computational model that's capable of using data from monotherapy cancer cell line screen. So these are the things readily available to us to predict clinical efficacy of untested drug combination. And we mostly care about that in patients. And one thing I want to point out in creating this model, having my pharmacy training background, we know drug concentration matters. So not only we have these in vitro drug screen done, we also care about, we only use those concentrations that's achievable in human body and looking for potential efficacious drug combination. So talking about computational model for drug combination, so very often people think about 2 factors, additivity and synergy. So these are the terms, just want to make sure everybody's are on the same page about these mechanisms. Additivity really refers to the combination effect. It is equal to the benefit of 2 drugs together. And synergy, we're talking about the combination of the drugs benefits are greater than the 2 drugs putting together. And for the most part, as of today, people has in the computational world pursue of synergy as sort of this the ultimate goal. Everybody is trying to come up with methods to different mechanisms, looking for most accurately predicting drug synergy. However, looking at literature, pretty much all the effort today focus on predict drug combination efficacy has focused on synergy. However, the performance of these models hasn't been great. This paper shown here, published last year in Nature Communication, represent one of the latest efforts in pursuing a computational method for synergy. What has happened is AstraZeneca has screened single drugs as well as combinations in a collection of cancer cells, And then they share these data both in the training data set and the testing data set through a crowdsourcing. So everyone who's interested in developing synergy as the method could join this dream challenge, and therefore utilizing your own method to predict drug synergy in these cancer cell lines. Long story short, even though many of the top methods was able to predict synergy pretty efficiently in the in-sample validation, which means you develop the model using the data set and making prediction using the same data set. When applied to outside samples, so basically develop the model in the training data set and predict in the testing data set, all the models fail. Basically, it's come down to about 50, I shouldn't say fail, but basically the accuracy in prediction is about 50%. Also, there's these early publications in nano-oncology has shown that through a meta-analysis, drug synergy method generated in using preclinical model was not able to predict clinical trial success or failure efficiently. So knowing that tons of effort in the synergy, so far they haven't turned out good results, we decided to build our model using a very different assumption, which is the independent drug action. So IDA, what does the IDA mean? So IDA, in a way, it's based on population. So what we're saying, when a multi-drug combination effect really is equal to the effect of the most effective single drug in these combinations. So we all know in drug development, drugs are tested in patient cohorts. So on the macro scale, patients are phenotypically heterogeneous. No patients are identical to the other person. Therefore, when you tested two drug combination in these population, one drug may work for a subset of population. The other drug may work for the other subtype of patient. So really, you see these two drugs together showing better effect. It's not because these two drugs have anything to do with each other, but rather the efficacy you see here is because every single patient in the cohort received more than one chance to respond to one drug. So this is not synergy, this is not additivity. So basically, it's just giving patients actual chance to respond to a therapy. And this is not only true in the macro scale when you talk about a trial cohort, a patient population. This also could be true in the macro scale. For example, we all know tumor shows high heterogeneity in tumor population. So a bulk tumor could represent a mix of different clones or different populations. So one subpopulation would respond to drug A, the other subpopulation could respond to drug B, therefore giving the two drugs together to this heterogeneous population, you see a better effect when the two drugs together. Again, the two drugs has nothing to do with each other. So based on that principle, this is how we set up our method. We have cell line data for hundreds of different drugs, so we can see, for example, in cell line 1, we see drug A have 90% viability, drug B has 40% viability. And if you take the IDA principle, so basically for this particular cell line, what is the best possible result from any given drug? That will be this 40% viability. And in cell line 2, again, these are measured drug sensitivity with drug A and drug B in cell line 2. Again, the best of the two are 70% viability. And then as a population, I'm simplifying this so you can take means of the population. So for drug A, the mean viability will be 80%. And for drug B, the average is 60%. And then if you take an average of through the IDA combo, this is 55%. So here you see that the 55% is 5% better than the best monotherapy. And so really the long story short is that even though individual cell line response to the combination are not better than the single best monotherapy for that cell line, when you take the average combination response, it is better than any of the monotherapy because the combination gave each cell line multiple chance to receive a drug that it may be sensitive to. So with this principle, the first thing we want to do, so we can do, and thinking about it, we don't just have 2 cell lines, we're dealing with a pool of samples with thousands of cell lines. So we come up with a method to be able to capture these best therapy response in each one of the lines and then calculate IDA combo. So the first thing we want to do is this truly present? Can we apply this to in vitro preclinical data sets? So we have identified 3 independent in vitro drug screen combination screen data sets. I mentioned NCI Almanac. So this is an effort by NCI. They screen about 5,000 drugs in 60 cancer cell lines, both alone as well as in combination. This is the dream challenge I sort of alluded to. Again, this one, all 3 study has looking at measured drug sensitivity, both alone as well as in combination. This last one is a Merck data set, screen about 600 drugs in about 40 cancer cell lines. So the dream challenge screen about 800 drugs in 50 cell lines. So the question we ask here is that for every single study, they have observed percent viability after combining 2 drugs together. We can, in our way, to use single drug treatment data using IDA combo to predict how would the population respond to those 2 drugs together based on IDA combo of IDA principle. So the very first thing we want to see, you can see is that most of our predictions are in line with the observed value. So which means the IDA assumption is true for a large number of drug combinations measured in these cell line pools. And so the Spearman correlation is all above 0.9, so 0.93, 0.96, and 0.93 here for all 3 data sets. In addition, just to see how far away from these diagonal lines, you can see about all 3 data sets, again, greater than 80% of the sample having the prediction error less than 10%. So really, we're quite, using IDA assumption, majority of the cell line measured drug combination efficacy fall into what we can predict. So even without the clinical setting, if you have data on single drug treatment, we will be able to pretty confidently project how the different single drugs could be combined together through the IDA assumption. But beyond the cell line, what we really care about, can we do this in patients? So what we did here is that we actually move into clinical trials. So first of all, to train our sample, we have tried to harmonize both CPRP and GDSC data, so that will allow us to have a bigger pool of candidate drugs to evaluate. In addition, we have looked through both clinicaltrials.gov and PubMed in order to select published clinical trials that measure both drug combination and single drug and compare whether the drug combination is better than the single drug in patient cohort. In addition, we have searched through literature to get at the plasma concentration of drug of interest, and then applying the IDA principle, we were able to generate predictive drug sensitivity combination efficacy. So what we did is to compare our predicted patient response in each trial to what's published by the trial. Here, the one tricky thing is we use power as a matrix. Because thinking about clinical trials, we can't simply just translate what we see, this magnitude of effect in vitro to patients. Every single clinical trial, the power depends on the number of events and the sample size, how many patients are in the trials. So therefore, and because these trials are done So we have a fixed outcome given certain power. So we decide to not using our sort of predicted in vitro data as an indicator of effect size, but plug into each trials with their existing event number and patient numbers in order to generate a power for the clinical trial. So long, sort of simplified way to say that is that the higher the power, the more likely we see a difference when compared to the combination therapy to single agent treatment. So this is the pipeline we use. We sift through, we don't want to pick and choose and have bias in the selection of clinical trials. Therefore, we use a method of both web scripting as well as manual inspection of literature. We start with over 22,000 clinical trial record and focus on cancer in phase 3 trials, and with all our drugs of interest indicated in the drug, we ended up having about 54 usable trials for our evaluation. This panels here really show the top 2 are patients in the trials that treatment naive patient. They haven't received previous therapy. And C and D are those patients who have undergone previous treatment. And then the A and C are indicate, we use the progression-free survivals from the trial, and B and D are those trials that we have overall survival information. So to walk you through this one figures, basically for every single clinical trials are indicated as a circle here in this graph. So if the trial decided the combination therapy is better than monotherapy, that will be true. So these are facts derived from the report of clinical trial. If the trial determined the drug effect, the combination is no better than single agent treatment, that will be in the false group. As I mentioned, we basically calculate the predicted power using both the projected the combination efficacy as well as the sample size and event rate from the clinical trial. So the higher the power we have, the more likely a trial will call the combination is better than single agent treatment. So using a very commonly accepted 80% power as a cutoff, you can see we can pretty accurately, so our accuracy is about 85% in predicting drugs that would be better combined in the cancer setting. And there are 4 clinical trials that was incorrectly classified. I can tell you 3 of them are ovarian cancer trials, and 2 of those trials actually, even though they're in ovarian cancer, so these 2, they were called, so the clinical trial called them the combination show no better than the single agent treatment. So these are carboplatin with paclitaxel, and we all know in treating ovarian cancer, these are current right now, they are standard of care treatment. So even though at the time those clinical trials defined the combination is no better than single agents, we still show that so that's part of the observation that we see. And so this is using progression-free as survival as the outcome from clinical trials. And this one is using overall survival as the outcome of the trials. You can see we have very high level of accuracy. We can predict, even though we're generating the model using in vitro cell data, we were able to accurately predict overall survival benefit in the drug combination with 93% accuracy. But keep in mind, the sample size here is very small. We only were able to find 3 trials that are showing the positive result for combination. Unfortunately, our analysis as of today do not do well in those patients who were previously treated with other agents. We can talk about possible explanation for this less ideal performance about the models. So just want to run through the, so those so far are proof of concept. We generate the model using these monotherapy in vitro drug screen data. We were able to recapitulate the finding pretty accurately, both in vitro and in clinical setting. And we think moving forward, we care about applying this method for prospective nomination of efficacious drug combination. So in this heat map grid, basically it's indicator, different color, darker blue indicating higher combination scores. So meaning the two drugs together will showing higher efficacy. So you can see, so drugs are listed on the slide, different, of course, the same drug combined with itself, based on the IDA principle, they should not be better. So they're all white. The black dots here showing just the missing data, and the darker the blue, the better the combination efficacy. You can see some of the grids shown here. These are drugs that basically the same type of drugs. So if they are all EGFR inhibitor or they're all MEK inhibitor, when they are the mechanism of action is the same, you see based on IDA principle, they do not help do better when combined. And then things we hear also is about, can we not say in the practice for certain disease, in this case lung cancer, we knew if we have a drug of interest, we want to develop another drug in combination with the drug. So this case is navitoclax. It's used to treat lung cancer. We want to know, can we add another drug to improve our efficacy? These are all different drugs that could have shown higher combination efficacy. Notice the number 2 and number 4 are both taxanes. So based on our model, taxane could improve navitoclax sensitivity, efficacy in lung cancer. And in fact, this finding was supported by several independent groups experiment both in vitro, in vivo, and in early phase 1 clinical trial that the combination do show better efficacy, and the combination can be safely administered to patients. And this is a 3D plot showing you, so navitoclax single drug alone are these. This is for docetaxel, that's docetaxel, and this is paclitaxel. This plane is showing best possible single agent efficacy, and the purple dots are based on IDA combination and what is the achievable combination efficacy. You can see these are significantly better than a single drug alone. And in fact, when people think about combination therapy, oftentimes one other concern is toxicity, but based on this big difference between the purple line and the single agent at best possible efficacy, we could potentially administer almost about a third of the concentration of initial concentration of drug, still achieve the same level of efficacy. So with that said, to conclude, we are developing novel computational tools, building using these in vitro drug screening data sets to enable drug sensitivity prediction, both along a single drug and combination. And when apply these to large clinical data sets, we can generate new large pharmacogenomic data sets for biomarker discovery and drug repurposing. And we hope, these tools could really enable discovery of new drugs and explore new mechanisms of action. So to finish, really, this is, I'm here to present the work from my group, and there's so many talented people in groups, everyone contributing significantly. I'm very grateful for my very positive circle of people, and they are the one make all this work fun and possible and exciting. And of course, the funding resources are great, and we have a large number of excellent collaborators, both within and outside the university. Thank you.
Well, thanks so much, Stephanie. So Anya and I will field questions. If there are any questions, please either type those into the Q&A module, or you can raise your hand. And it looks like we do have a few raised hands already. So we're going to go with Dr. Yi first. Dr. Yi, you're allowed to talk. Go ahead and unmute.
I hope other people that were in line, I didn't jump the line here, but at any rate, Stephanie, that was great. And I wanted to ask you kind of two semi-related questions, or maybe not. Can you use this technique to model host toxicity to try to expand therapeutic index and really look at drug efficacy that way? And the second part of the question is how you think about modeling antibody drug conjugates. You know, obviously they have two separate functions. It's a variation on your combination data, but I wonder if you've been thinking about that, given that at least in breast cancer, that seems to be where the field is going at the moment.
Yeah, those are excellent questions. Those, to be honest, I haven't thought about it much. On toxicity and especially on the combination end, we got asked quite a bit, you know, about the, you know, doing with combination in general, you will have higher efficacy, but probably suffering in the toxicity end. So our straight answer there is to, you know, that example I'm showing that you could potentially lower the single agent concentration, but that's not what you're asking. So on the screening for toxicity, I mean, the principle right now, I believe in these mechanisms really do exist. So it's come down to what training data set we use to answer the question. In my earlier in my career, we have utilized lymphoblastoid cell lines. So those are normal cell lines, B cells, and we have actually tried to use it as an interpreter of sort of toxicity interpreter. And I know others now has generated iPSC cells in order to differentiate into tissue of. So again, the principle, our method will, you know, we can apply if there's any screen of compounds. So far we built our model based on cancer cell line screen. If there's any drug screen in normal tissue or normal, you know, that could be used. You know, exactly same pipeline, just apply to different data. We can use that to address some of the potential toxicity issues. Regarding the conjugate, honestly, we haven't done much. Mainly, you know, sort of what's conjugated is a little tricky because it requires that sort of almost you need the microenvironment, you need the system there to be able to, because there is a delivery efficacy and that issue, not simply just the effect. But one thing recently we're trying to expand our model because so far everything we used were in vitro cell line screen. We have started some recent collaboration with people who develop model as organoids and also in PDX. So we're hoping they are doing high throughput screening in organoid and PDX. We are right now trying to set up collaboration to work with them to you know, build our model in those training data sets, because that in a way is a little better, closer from cell line to the individual. We want to compare, do those have better, you know, so long story short, the conjugate, I think to screen and cell line make no sense to me, but if you test the conjugate in let's say a PDX model, that makes sense to me. So that we're not quite there yet. They are screening tons of different, so we are working with people in Jackson Lab and also Brian Long from the University of Utah. So they both are developing different organoid PDX pool and for high throughput drug screening. So we're aiming at team up with them to explore our tool can that be used in these other models.
Okay, thanks.
It looks like we have two more questions. We'll start with Dr. Van Ness. Dr. Van Ness, I'm going to allow you to talk. Go ahead and unmute. You're able to do that? Okay, we'll come back to.
Wait a minute, am I in now?
There we go, gotcha.
There you go.
Go ahead.
Okay, great talk, Stephanie. You're always inspiring new ideas. One of the questions I have is, can you refine your combination therapies by trying to do imputation from single cell analysis where you can actually identify subpopulations within a tumor? Therefore, the single cell analysis will give you a real readout of the heterogeneity and then impute which are the best drugs to hit the subpopulations.
That's exactly what we're planning on doing next step, because when we started this project, getting a clinical data on the sort of on population scale, the whole reason the idea worked is based on this population. Because, early on, even myself are confused, there's synthetic lethal, to me, that's really in the same body, same tumor, how the two, hitting
Correct,
these things to work. And this IDA combo is really different than that. It's working in the population. The population has to be heterogeneous. So I think when we start the work, clinical trial is a natural target to start working on. Right now, with single cells become more readily available, we are working with several investigators. Dr. Clark Chen is one of the examples. We're thinking because they have single cell sequencing RNA data. So we are in the process of trying this out if we can impute in every single cell for potential drug response, and then we treat the bulk tumor in several different ways that inhibit different forms of would carry different sensitivity. So that's exactly the goal and the next step that we're aiming at doing. And also in my lab right now, we're trying to create experimental system that, testing single drug, you can expose it to a cell line, that's fine. But IDA require a population. So now we're creating these pooled samples. So we're mixing cancer cell lines together to create these potential cancer population in order to test IDA combination in the cell model.
Okay, that's great. Thank you. We'll be talking again Thursday, so we can expand that discussion. Thank you.
Yeah.
Dr. Belinsky, do you want to read off some of these Q&A questions in the Q&A module? Otherwise, I can do it too.
Sure, I can do that. Is Scott, I don't know whether Scott is online. Scott, do you want to ask? Okay, so Scott Dame sent a question. Are there any limitations of using gene expression data for imputing drug sensitivity? And is there potential for improvement if proteomic and/or metabolomic data were available for tumors and cell lines?
I think the short answer to that really is yes. I think I do, I have a lot of work early on in my career focused on DNA end of things. And I've been telling people I'm a converter to RNA because I realized for prediction per se, DNA, even though the signal is correct, because the sheer amount of the scale, you're dealing with billions. So single effect, even though real, that just predict in the clinical prediction, they're never sufficient, at least till now. I haven't seen that being very, things are not packaged together in a very accurate way to predict phenotype. I think the reason we focus on expression is because the technology is quite mature now, and we were able to generate prediction fairly straightforward and with. But I do believe, proteome and metabolome are even closer from expression to the ultimate phenotype. Therefore, they have a lot of potential. My only concern is sort of technologically, as of now, are we ready there? Because we have tried to work with some protein level data. For the most part, there's various methods. LC-mass spec tend to be slower. Costly. There's always that cost. So we haven't had the large enough data set. You also require large enough training data set to have that robust power to move forward. So I think the limitation is more in the technology rather than the biology or the principle behind it.
Yeah, but along those same lines, and Haideng has also a follow-up question that he can either ask himself or I can read it. But I was wondering, one of the things that I love about your approach is that it's very powerful in uncovering new biology. And are you using at all genetic interaction data sets? They are mostly not available for exactly the cell type that you're looking at or that you're studying. But there is really a growing market of genetic interaction data.
Yeah, I think that's true. It sort of get back to our expression level right now. It's very simple model we have. We don't have much interaction. We have the expression value. People I think in the genetic world, there's better way to, using pathway or using interaction network to represent different modules to predict things. The tricky thing there is, you know, on the bioinformatics per se, you can always say you run a pathway analysis or identify some module. But the difficulty is to translate that directly to the true mechanism or to something you can really use.
Yeah.
So we sort of resort into at least the gene-based, like expression. Yeah, it's simple, but you can really point at this gene has a role rather than this module or this network or this pathway has a role. And then it's a little, I don't think we have a great method right now to pinpoint in that module what really drives the signal. So yeah, I think we are thinking about both approach. In general, we try both sort of single gene, and we also do, sort of other machine learning. One way you can take sort of almost like a tag gene out of the whole transcriptome, pull out just about 200 genes that could represent the whole transcriptome. But then, those are just by association. They may not be having any function.
Right. But at least you get a hint, right, of the networks that might be participating.
And then we also learn from our experience of repurposing, a lot of drugs that we think we know what they're targeting.
Right.
It really isn't.
Right, exactly. Yeah. So Haideng Ren had a similar question. He was specifically asking, is there any correlation between drug sensitivity and variant allele frequency for tumors and cell lines?
That's a good question. I don't know that yet right now, mainly because we have done similar work sort of related to toxicity in germline tissue. So yeah, in there, I can honestly say yes, drug sensitivity and germline genetic variants do associate, and oftentimes it is associated by genetic signal regulating expression. There lead to drug sensitivity difference. So that we can do. In tumor or in cancer cell line is a lot trickier because the mutation profile is very different. So in germline, you have your SNP that has hitting at least 1% in the population frequency, versus in tumor, there's so many sporadic mutations. So, and we all know we can't just, the bioinformatic tool is to call that gene. You can count how many mutations there is. Therefore, that gene is important. But the biologist side of me knows some mutation will lead to better outcome, others will not be. So simply to throw them in a pool to call that gene important, it's not ideal. So we have tried some other way, looking at sort of compared to sort of if you have a random frequency of mutation, only call those mutations above the random frequency backline. Those are the potential functional mutation, but still, it's still at the level of counting number. And even with TCGA of 10,000 sample, with that kind of mutation profile, I don't think we have any power to truly say anything between the mutation and the, with exception, some we do. That's given the mutation happened at a higher frequency that was able to, we can use it. We have, not necessarily in this setting, but we have separately when looking at biomarkers, we have spent a lot of time focused on long non-coding RNA. So in that sense, we have found certain mutations. Say in GWAS, we find SNPs that's in the inorganic region or not in the exonsonic region. We have found those SNPs are actually overlaid with long non-coding RNA, potentially regulating the expression of long non-coding RNA and lead to drug sensitivity. So we have examples of those. I don't know, I would confidently say we have comprehensively studied that.
Okay, thank you so much. Are there any other questions? Okay, then I want to thank Stephanie for taking the time to present the data here today. I want to thank everybody who participated in a wonderful discussions. Thank you and take care, everybody.
Thank you very much.